Right. Thank you for having me. I mean, it's a real privilege, and I really want to thank all the organizers for this opportunity. I mean, when you see the list of speakers, that also the ones that preceded me, and now the ones that come next, it's really an honor to be here, and a great opportunity. Also, I particularly liked when Nicolas explained to me what this was about, the fact of being in contact with a very young audience, and then based on that, I decided to rather than give more of a scientific talk, explain a little bit my career, maybe to give you, hopefully, to give you some insights, for young scientists, where do you want to go? Yeah, hopefully that, see if that works. Yeah. So maybe just as a way of introduction, I thought I would actually say, give you an overview of my career, because it's not a straight path, right? And that is what is, for the good and the bad, is unique about me. Maybe not unique, but not rather exceptional, right? I, like everybody else, I started at the university doing the PhD, right? And this was at the University of Barcelona. But I already then, I had the opportunity of for going abroad. Once at Zurich with Professor Van Gunsteren, and another 6 months with Professor Tak Koens. For those of you who don't know, he was the first to publish about docking methods, like back in 1982 or something like that. So almost 20 years later, I went to his lab. And that was a terrific eye opener for me, both stays. So I think this geographic mobility and cultural mobility also is something that, I mean, most of us experience, but it's like really a good opportunity in science, right? And then after my PhD, which I was very happy about, um, but I also decided academia is not for me. So actually, I didn't go for a postdoc. I was lucky enough, and this I have. I mean, we have to acknowledge times have changed, right? There has been since the start of my career till now, lots of ups and downs in the biotech pharma world, and now it's not a good time. But back then, um, you could finish a PhD and after that go into in this case it was biotech, where Bernales, which I'm sure you heard about, actually, amongst other things, because Nicolas Golop was there already when I started, and I had there a few very, very nice years, 4 wonderful years. I really enjoyed that. That was a structure-based drug discovery. I was really lucky to start with a project on a protein, Hitchcock protein 90, which went all the way from just starting virtual screening and other things. We did fragment screening, and finally got to candidate, which was our license. This went to Novartis, and it went up to phase 2 clinical trials. It finally failed. We can talk about that maybe, but it was really a great thing. I really enjoyed that. And but you've met people from Italy or Spain or maybe other parts of the south of Europe. You know that we always have this deep roots. So we had 2 kids actually while living in the UK, in Cambridge. That was a really very nice experience all around. But we wanted to raise them in our, in Catalonia, where we are, both my wife and I are native from, and that was the main reason why. Then I was looking for opportunities to go back, and something happened that was unexpected. I had the opportunity of becoming a career research professor. Again, this is not probably the easy nowadays, but back then it was relatively easy for me. I had published during my PhD and also during the time I was at Bernales, so I got, I guess, lucky and got offered that opportunity. And then I had to transition back to academia. And that was a difficult thing to do because I really liked applied drug discovery. So creating a group from scratch, etc., that took some time. Not straightforward. But my experience actually was really helpful in many ways, and one of them is identifying where I wanted to work. So before, when you're doing the PhD, you think the computational methods are wonderful, and then you apply them in practice, and you find that they're failing all the time, or not sufficiently good, and that gives you a lot of motivation to then go into the basics and try to improve things. So I was working on things like trackability predictions, improving virtual screening, and other aspects more fundamental, and so on. And this is what I did for almost like 18 years. And during that period, also some of the things I did in academia, then I had the opportunity of translating into companies. Minorix first, then Gain Therapeutics, and more recently, Oniria Therapeutics. And in all cases, these were companies created to discover drugs for, in Minorix cases, for rare diseases. Gain Therapeutics, in the end, that was for Alzheimer, and Oniria in oncology. And then in the year 2023, I was like, what do I want to do with the rest of my life? Do I want to stay in academia until I retire, or and then I started looking for opportunities in pharma, because I wanted to do drug discovery, but with just focusing on that rather than the multitasking that you will see involve being in academia and also being in several biotechs. And I'm very happy about the change. It's now been over 2 years, and it's a different world, but it's also nice. And that's what I will try to cover now for the rest of the talk. What I will try to do is I define 3 dimensions that for me are important: the goals that you have in that organization, the freedom that the individual has in that organization, and also what happens with the rest of your life, right? The lifestyle. And then I've tried to then go for each one of these, academia, industry, and more of a startup environment, so that you can get a feel for, maybe where do you think you would fit more, right? Right. So academia, I would say, this just to make it a bit lighter, I always have a cartoon here. And here this professor says, I can't come for a drink, I have to finish this. It's publish or perish these days, right, in academia. And the other professor says, yeah, we seem to be on an efficiency drive. We're doing both at once. And that sums it up, I think. In academia, the main goal is to publish. And I think that's, to me, that's why I always was a bit uncomfortable in academia, because the people who are really happy in academia, I find, are the ones that when they publish a paper, they get a kick out of it, right? They really are very happy of having published. And to me, often, like, the discovery process itself was super nice. I really enjoyed that. But then actually all the work involved in publishing is something that I didn't enjoy much. But actually, to get to publish, you have to do many more things, right? You have to think, have your own ideas, and then turn an idea into a project, design that project, how it's going to be done, get funding for that. Actually, writing grants, for me, I mean, many people hate it, but for me, writing grants was actually kind of nice, because it's like you dreaming of possibilities, and then it forces you to think how you're going to achieve your goals. So that was good. And the part that I really, really enjoyed is having a team of very young, mostly PhD students. That was, it's a very youthful and positive environment, and I really enjoyed that. On top of that, you also have to build collaborations, because obviously nowadays everybody does, fortunately, collaborative and multidisciplinary research. So you have to meet people, share interest, find where you can collaborate, etc., etc. Some people, I didn't have to do teaching. Some people, obviously in academia, most people do, and there are a bunch of other things, right, that go around. So yeah, it's very flexible in this way. There's many things. You have to be a bit of a one-man orchestra, but publishing is like the core of it, right? So you definitely have to like that. Otherwise, yeah. And on that note, I because that's really what I enjoyed the most. The people I supervised during their PhD or the postdoc and master students, and also the working with. So we had some very good relationship with long-standing collaborations with other groups. This is not an exhaustive list for the collaborations. It is for the former people in my lab, and I really enjoyed it, and I want to thank them for all those years. That was a real privilege. Right, then we move to another dimension here, which is freedom. Right, academia is supposed to be like the out of the three environments, the one that where you have more freedom. Right. I'm sure you're familiar with the PhD comics. And this is actually right. So yeah, I am going to research whatever I want, but first you have priorities, right? You have to do whatever your PhD supervisor tells you. Then you have to research on whatever is going to give you publications, whatever is going to give you grants and maintain the group. And then by the time you get your freedom, maybe it's already a bit late. But of course, I mean, part of it is true, right? But it's also depending on how you take it. And indeed, the freedom is there. And actually, for me, one of the great things there is this opportunity of having projects that are like long term, right? So I'll give you an example of something that I managed to publish only recently, like a few months ago. But this started, I don't know, easily 6-7 years ago, and it's I'm quite proud of it because of several aspects that really summarize what it means to be in academia in my view. It's a problem that other people don't have the luxury to investigate, right? So in this case, what I observed is that BAT4, bromodomain, a protein that is a very well-known target with compounds in phase 2, phase 3 for oncology. Lots of compounds have been developed. There's all the structural information that you can have on it, etc. Everybody knows that there's here where I'm pointing, all the ligands put something hydrophobic here, right? And everybody knows that this methyl here is actually in direct contact with 3 water molecules that are part of a network. And nobody seems to care. They design the molecules and then they find that the optimal substituent here is something hydrophobic and they go with it, right? But I was like, this doesn't make sense. You have a water network and this is hydrophobic. How come? This doesn't make sense. There were studies with people trying to displace the everybody was discussing about I'm getting a call or something. Sorry. Are you still there?
Yes, we are.
Okay. I was getting like a call or something. I don't know what. Sorry, I don't know what happened.
You're sometimes breaking off a little bit in terms of sound for me as well, but most of the time it's okay. So I think we just continue, and if hopefully you can use as much of the bandwidth as you can.
Okay, good. All right, then. So I was saying that I saw that there were studies with people trying to displace these waters. This is something that is everybody's minds, right? You see a water molecule between a protein and a ligand, you think if you can displace it. There's been a lot of studies on many proteins, and then for this protein also people were doing that. But I was astonished, like nobody cared that this is completely unnatural, right? In principle, you have something hydrophobic next to a chain of waters. So to address that, then we established a collaboration with synthetic chemists who made modifications to that position, making also polar groups. And we established collaboration with a group of Frank von Delft in Oxford, where they did crystallography, and we also did biophysics in collaboration with Carlos Galdeano in Barcelona, and obviously computational chemistry. And in the end, turns out that actually, yes, these waters create a hydrophobic environment, and actually, I think now that this not a unique case. It's like a recognition motif, a molecular recognition motif that has simply been ignored before. And maybe I give you my 1 minute summary of why this happens. You may remember from school that the hydrophobic effect is an entropic one. So waters enthalpically are not unhappy being around a hydrophobic area. The problem is they have to organize and form all the hydrogen bonds between them to then be able to be in contact with something hydrophobic. Well, what happens here is that the protein is taking care of pre-organizing these water molecules. So when you put something hydrophobic here, it's actually favorable because that hydrophobic group, when it was exposed, it was creating order on the waters that were around. Now, when you put it here, because the waters are pre-organized by the protein, actually, you're not paying that entropic cost. And then, in fact, because you put something hydrophobic here, you're facilitating or reinforcing the hydrogen bonds of those waters with themselves and also with the protein. So you're making a much more tight, much more compact network of hydrogen bonds. And then the protein actually sort of collapses and reduces its mobility. So that, when we finally got to understand that, right, this aha moment, right, that was super nice. Then after that, there were a few years of writing the paper and everything. That's what I was saying. I didn't enjoy that particularly, but it's a privilege to actually have the time to investigate something that is not of immediate impact, right? And this is what academia provides, and it's actually really nice. So if you want to know more about that, please go ahead and read that paper. It's one of the papers, of the few papers that I'm really proud about. Okay, so lifestyle. Yeah, we all know that professors don't make as much money as people in industry. So at least probably you have a better lifestyle, you would think, and the reality again is probably not that glorious. As you take more and more responsibilities as a professor, you spend more and more time in meetings, organizing things, and less time for science. And the day simply doesn't cover, right? You cannot get everything done in the daytime. So you become a bit, overstretched. You tend to reach many things at the same time. It's can be difficult to manage as much as in anywhere else. But it's also true that actually you're your own boss, right? So you have time for whatever you want to do, right? So probably you will not have a lot of spare time, but you have a bit of freedom of where you put your time. And for me, for instance, this opportunity of having this, side hustles with, investing time in creating companies, spin-offs from the university, that for me was like super nice. It was obviously didn't help with the work-life balance, but it really was a good thing to do for me. Other people do consulting. Other people maybe they are a bit more careful to keep their own private time but yeah, that in academia in general you have this freedom to decide where you want to invest more of your time. Some people go into more management, so there's a lot of opportunities there as well. Yeah? So that's that covers academia for me. So now we go for startups, and here it's very clear, right? So you want to make money, and you want to make money fast. That's the theory anyway. In reality, obviously, what you have to do is to find which product you're going to create, and you have to then convince other people to invest in helping you create that product, and you have to also convince people to attract people to your team, that they should be very motivated also on your objective. You have to do a little bit of everything. You have to negotiate. You have to do discussing with other companies, potential partners, right? So all this is part of the life. It goes much beyond science, etc. Right? And then hopefully, and maybe who knows, you may also get some return. The reality is that this plan of making money fast really doesn't agree very well with the business of drug discovery. I'll explain why. So this is the first company I was involved in the creation. We founded it in 2011, right? And now it's 2026. We are in phase 3 with a compound that was actually we had the patent in 2014, so we're quite fast here. It's a repositioning project, so it's not we didn't have to discover the molecule, but discovered that it could be like really useful for particular indications in rare diseases. And since 2014 till now, it's still in development, and nobody's buying. Actually, I have a one of my the same process, and nobody was buying. And then one day he started getting calls from companies saying, "Okay, we want to buy you." And then because they knew that the FDA had approved the drug before he did, and then as soon as the drug was approved, then they wanted to buy, right? But many pharma companies also don't want risk. So nowadays, they are happy to let biotech companies just go ahead, take all the risk, and then eventually at the end, then buy the asset. Obviously at a higher price, but they can calculate exactly the margins that we will have, etc. So then for biotech, it becomes a very, very long journey. And this, it's also something that is difficult. First, for someone like me, very interested in the beginning, in the discovery process, etc. Then later, you go into regulatory, clinical trials. It's a different type of involvement that is needed, right? And the same pattern has been reproduced then in the other companies that I co-founded, Gain Therapeutics, which actually was like a spin-off of Minorix Therapeutics. It's now in phase 1B in clinical trials for Parkinson's disease. But yeah, still a long road ahead. And Oniria Therapeutics, that was in oncology. It was a really nice long collaboration we had with an academic group in oncology in Barcelona, in the Vall d'Hebron Institute of Oncology. And eventually then they had a very interesting target. We identified allosteric agonist of that molecule, and then trying to understand how this can help in which cancer, and also optimizing the molecule, etc., etc. And then, once a candidate was selected, that's also when I moved out of the company and moved into Boehringer Ingelheim. But again, long road ahead. So, I mean, there will be still some business cases that maybe you can generate the product in a short time. Maybe you can expect to get a return. But if you really want to develop molecules, prepare yourself for a very long trip. And of course, what's happening on all these cases is first, the companies have a lot of emphasis on the platform, on the technology to discover molecules. Then when they have molecules, then they have to put all the resources in paying for clinical trials. And then oftentimes they then abandon the technology because they have to concentrate. They cannot just generate more and more molecules, because every time that they develop a molecule is like a commitment for many years into clinical trials, right? Yeah, and then also like in Gain and Oniria, these are first-in-class drugs, which also doesn't help. It makes it even more difficult. You have to open a lot of new understanding in terms of the biology, etc., etc. Right? So yeah, it's a very exciting. I really enjoyed it. But yeah, the expectations, right? Just to manage expectations, it takes decades. And this is something that I certainly didn't expect when I started back in 2011. Yeah, freedom. As long as your idea, if somebody's willing to sell it, any idea that you can have, if there's a buyer, it's a good thing, right? So I don't know if you know Calvin and Hobbes. It's my favorite comic strip, and it's always very funny. So this guy is very entrepreneur. He has lots of ideas of what he can sell. I don't think he has many buyers, sorry. But yeah, it's the same thing. As long as you can imagine something, often people start with an idea that they think there would be a lot of interest, and shortly they realize, well, actually nobody wants to buy it, right? Why nobody would want to buy scientific names? It's anybody's question. But you have to then, the first thing you have to do is go into the market and see if there is an interest. That's the degree of freedom that you have. Then once you have decided on your product, then everything should be focused on that. So you have to be aligned with that vision of the company, right? So for me, again, it was about the technology that I had developed in the university. It enabled us to rationally discover allosteric molecules, and I wanted to apply this at a bigger scale. And I could do that through minority therapeutics and gain therapeutics, and these are examples of different projects where, using the technology from my group, we discovered allosteric pockets on the protein, and then we did virtual screening and identified molecules that bind to those proteins. And here, because it was mainly initially rare diseases, the point is these molecules don't need to do anything functionally. All they have to do is to bind to the protein and just keep the protein folded. Because these in rare diseases, you have a mutation where the protein then becomes less stable. And because it's less stable, then usually even at the ER level, it's the quality control mechanisms in the cell recognizes the protein as misfolded and remove it. So if you can bind to the protein and keep it a bit better folded, then what happens is they can reach the target organelle. The majority of these cases, these are lysosomal proteins. And then you recover the function. And if you recover the function, then you should have a very direct link in this case between the target and the phenotype, right? Because of these rare diseases are mutations that are a single pole mutation is causing a whole organ disease. So very, very happy about that. And this is the project that is now in phase 1B clinical trials. The others, I don't know what stage, but we actually published about them. So it was a good way for me. It's one of my motivation for me was to be able to try and demonstrate that the technology was working. Right, so much for freedom. What about lifestyle? Well, if you like living, interesting life, then definitely a startup is your thing. You have to, you can not wait. Sometimes you have you have the opportunity of making a partnership with a big company, and then you have to see, how you adapt, how you find the resources to actually demonstrate that the company that yes, indeed, you can do something interesting. Because having a deal, if you're a small biotech and you can make a deal with a pharma, for instance, it's a big thing, right? So then you have to adapt to that. You shift all the resources immediately there, and that creates a lot of tension in the company, but it's something worthwhile. Work-life balance, well, it also is not ideal, I would say. Obviously, then you say, I made this. It took me many years, a big effort. A lot of people had to contribute to that. Many, and then the valuations that you can get sometimes are disappointing. There's lots of things like this. But yeah, I mean, the other things that is actually quite nice about the startup is obviously you can get some stock options and equity. But also, one thing that I really like already back in the time when it was at Bernales is the opportunity of really seeing the whole company, right? It's much smaller than a big pharma, but still much bigger than what you see in academia, and then you get to know everybody in all the other functions of the company, not just in the scientific functions, but also business development, the CEO, everything. And that's also nice. It gives you more of a holistic picture of what a company is, right? So that if you enjoy that, that's definitely a good selling point for a startup or a small biotech instead of a bigger company. Yeah, and sometimes you get this very good, nice moment. Just fun fact, if you look at here, you try to find me, you will see I'm not here. And the reason is because we actually managed to, so we did the NASDAQ listing for GAIN during COVID. And so this didn't happen in COVID, right? Nobody could travel or anything like that. So 2 years later, they organized a trip to New York just to take that picture. And I said, you know what, I don't want to go there. And I think I did the right thing because one of my colleagues then actually got COVID during the trip, and then she had to stay in a hotel in New York for 4 weeks until she was then negative again, which was not the nicest experience in her life. So anyway, but it's a fun anecdote. Right, industry, and here I'm changing to Dilbert because it really is in the spirit of bigger companies, right? It's a bit cynical, and Dilbert, this is Dilbert's was says, this week I achieved unprecedented levels of unverifiable productivity. I made phone calls, built consensus, displayed leadership, attended meetings, and set priorities. Right. That's what, in a nutshell, summarizes some of the work that goes into big organizations. Of course, this is a bit of cynical, but actually, companies are created to deliver on a product, and because they are big organizations, you really need to coordinate and to communicate across departments, which can be a challenge, but also is something very nice. And actually, I would argue, big pharma is the only entities that are really able to put the investment and the commitment to take a drug into the market. The majority, I mean, you've seen a picture like this or similar. This is provided by Novartis, 2.6 billion cost, but it's just the time that it takes, 10 to 15 years. My company, which is a medium-sized pharma, last year we're super happy because 2 compounds actually reached the market after many years. Songertinib and nerandomilast. And you really need this sort of organizations if you want to actually reach the patients. So it is not just about the science. It's really, you need to create the whole machinery and the resources and the power to actually, commit to something for many, many years, knowing that 95% of the projects actually will fail. And you will have to then get one that is successful that then continues enabling all the discovery, etc. Right. So this is absolutely fundamental function of pharma companies. Freedom. Continuing with Dilbert, this is a photographer who used to have Javier working independently, and he was completely unsupervised before, and now he's in a company, and he has to fill his time in 15 minutes increments, right? You have a document where you have to say what you did in which project you contributed, and in this case, every 15 minutes. And the fun thing is that they even have a code for people who try to strangulate themselves, and if you succeed, the code is different, right? So that's again a bit cynical, but yeah, it's a bit more being like a clock in a machine. That's the typical stereotypical view. Some of it may be true, but actually, what I find is that well, you don't have the freedom to choose in your projects. For instance, you really can enjoy working in projects because I will maybe say a bit more later. Both projects end up being very big and have all the functions and all the people that they need. And yes, this is complex. This is I want to acknowledge here the image taken from a webpage of Catapult Medicines, a consulting firm in the UK. Drug discovery is in itself is not only very unique but also very complex, and you need very different knowledge areas. And working here at the eye, for me, is the first time where we have absolute experts on every single aspect of drug discovery, right? So pharmacodynamics, pharmacokinetics, toxicity, you name it. Everyone. I mean, and this is fantastic. Having all these things together, put together for the purpose of actually developing a drug. Of course, there's a risk that because you create this that people may work in silos. All these things is also natural for a big organization, but actually, it's quite it's very, very nice to have experts on every single thing, and then they are there exactly for that purpose, right? So it's the natural environment of pharma companies, right? And lifestyle. Robert Dilber says right here is work-life balance. We shouldn't even use that term, right? We can call it work-life integration. And then if you don't have a life, then so much better for the company. Well, that's definitely very cynical. I would say that compared to the other environments is where you can, if you want, have a better work-life balance. Also, if you're ambitious and you want to spend more time, then you're welcome to do that. Also, I would say on the biotech depends, but compared to academia, you also get better salaries, and you can have other benefits like bonuses, and then you have opportunities for also changing career. If you want, even within the company, you can go into business development or licensing or more organizational, like just aspects. So there's a lot of possibilities in a bigger company as well. So that's all. So I would say here, it's about yourself, what you like, and also this can change during your lifetime, right? So if you don't worry. I mean, you start obviously sometimes in the beginning you cannot choose, or maybe you will be able to choose, but then maybe 10 years later you say, actually, I want to try something different. So that's what I would like to encourage you to think in these terms. You don't have to have a fixed path that you have started and then you will have to follow all your life. There's freedom. It's not easy to change, I have to say. People don't understand that you may want to have an atypical career, and that's all right. That you don't fit into what people expect sometimes, and you may have to pay a little bit of price for that. But it's about you. So try to build your own career. And here's so it's a bit, actually in my view, the three environments are complementary, and they actually sort of fit each other, right? Academia obviously more on the basic aspects of understanding the biology, developing new methods, etc. Biotech trying to capture this transition in value, but also the highest risk part, and industry really creating the environment necessary and the commitment, the stamina, the resources to do this very, very expensive clinical trials. I think that's the natural division for me, and then all they all fit together in this way, and you may be interested in changing stages during your lifetime. Final recommendation: have fun. And by fun, I don't mean that it's easy. I mean that you enjoy it. Yeah, and this is Calvin again. It's a very very nice one where he's digging a hole and he's all making all this effort to dig a hole, and Hobbes asks, "What have you found?" He says, "A few dirty rocks, weird root, and some disgusting grubs." And he's really impressed, and his friend is really impressed. In your first try, yeah, there's treasure everywhere. So if you enjoy digging out disgusting grubs, there's nothing better, right? So follow your heart, and there will be treasures. That's I guess my message. And also, don't forget, you will be always working with people. Treat them well, make friends along the way. And yeah, I want to thank obviously all the companies that I have been involved with, and all of you for your attention. And please, if there's any question, I will be really happy to answer them.